genetics lab, and we're working to extract DNA from the Castanea plant species, and there's two that we're working on, is Castanea affinis and Castanea sessiflora. So, do you want to talk about why extract?
Okay, so our purpose is to determine whether to this species have the same characteristics, because they're like two different colors, that is yellow and red. So we want to know like if they possess the same if they're like genetically related, or like they possess the same characteristics. So like to do this, we have to like clean up the seed, and with the use of some chemicals, chloroform,
There's mercaptoethanol and CTAB buffers. There's isopropyl and chloroform, which is my favorite.
and while performing this experiment, it is important to know that labeling can never be too much or less. So like we have to like take hold of what we are doing in labeling, because like we can.
It's easy to mess up and
Yeah, to
confuse the
mess up.
chemicals you're working with. So there's 4 main stages. There's preparation, and then there's precipitation, and there's cleaning, and then the nano dropping, which is once you get through all the cleaning and separating the contaminated DNAs, like contaminated particles from the plant sample and the actual DNA, you can go to make a gel, which is what you use to nano drop it, and that's how you measure. So just like, this isn't an actual, but this is what nano dropping, this image right here, this is how it would look. And if it goes into negative numbers in a specific column, they're hard to pronounce, so like, they're just, if it goes into a negative column, it's probably that you didn't clean it well enough, or you still have some contaminated plant samples within it. So it doesn't mean that you failed on it, it just means that there could be possibility of DNA. It's just how well you clean it and how well your process is.
So like in the it is okay to mess up while performing the experiment, but like you have to like correct the mistakes immediately, or else like you're gonna like mess up everything during the experiment. So that is a centrifuge, and it is used for mixing up the solutions.
It's basically a spinner that just separates everything into test tubes that you know.
And Castanea affinis can be found in California along the side road, and it is rare in Chicago because
That's the affinis, the red one. The red is affinis.
Any question?
Comment is just that you did a good job explaining DNA extraction, because I've heard that a few times, and when you guys have barely started.
Yeah.
So I thought you did a really good job first. It was fairly easy to understand. The question though is, why would anybody care if the red and the yellow flower are genetically related? Like, why are they researching?
Because the flowers are basically the same thing but different colors, so they're trying to know what is the cause for the flowers having two different colors and why not the same colors.
And I guess to maybe like, just to foreshadow, I guess, if they're basically the same species that have different colors, like if one is endangered, maybe you can like try to figure out what the reason would be, and to maybe protect the other species from going endangered or being threatened. So I guess, yeah, it's just that Laney's.
Did you guys ever hear anything about pollen, that different pollinators pollinate the different colors or something?
Well, from my own, I know that like animals or like insects, they go on their color instinct, like butterflies, that's sort of the colors they go, but we haven't really talked to them a lot about that. We just